So good evening everyone. Today I'm going to tell you all about gamma oscillations, and everything that we do in our lab revolves around these oscillations. So I'm at the Center for Neuroscience. I actually did my PhD at Johns Hopkins, and I'm delighted to see one of my advisors, Ernst Niebuhr, here. It was a long time ago. So before because towards the end things may, I may not be able to finish, so I'll just acknowledge the people who have done all the work. So these are the current PhD students, the ones listed in red. I'm going to talk about their work today, and collaborators and funding from India Alliance. So I acknowledge that. So we are going to talk about brain rhythms. Gamma being one of many different oscillations that we can record simply by actually putting electrodes on the scalp and getting a signal like EEG, which shows oscillations under certain circumstances. So gamma is one of many different waves. The one that you are seeing here is alpha wave. It's a wave that's very prominent when your eyes are closed but you are awake and relaxed. And then there is different waves like delta waves, theta, alpha, beta, gamma, and so on. They are all associated with different cognitive states. And you can see that gamma is associated with really cool sounding words. I picked this up from a web page called Health and Focus, and you can see gamma is expanded consciousness, whatever that means. But it is true that it is linked to high level cognition. So in meditators, for example, if you record EEG while they are meditating, there are reports that the gamma waves that you get, those linked to, by the way, again, consciousness, attention, learning and memory, it's really huge, never reported before in neuroscience literature. So gamma waves are huge in meditators. So I'm going to actually tell you a little bit about meditation data as well today. So let's get started. I'm going to actually talk about, half of my talk is going to be on EEG data, half of it is going to be on monkey data, but it's all going to tie up together with, everything is revolving around these gamma waves. So let me just tell you the type of plots that I'm going to show. So generally you can show a waveform as a function of time. So this is x-axis is time, y-axis is voltage here. And as you can see that this is a signal which has a prominent oscillation. In this case it's an alpha oscillation. So we convert it to what is called a power spectral density, where we are just plotting the power of the signal as a function of frequency on the x-axis now. And in these plots you get a nice bump at around 10 hertz or so, which means there's more power at around 10 hertz in this signal. This is the power spectral density. We also have what is called a time frequency power spectrum, where you show the power both as a function of time and frequency, in which you see this alpha wave as a nice horizontal bright band here. Brain signals often don't have any strong rhythmic component. So you often will have signal that looks like that, and there the power as a function of frequency shows this fall off. This is true for all biological signals. There's more power at low frequencies, then it falls off, but there is no bump in the power spectral density. Okay, so there's no rhythm in this signal, and then the time frequency plot looks like this. So let me just show you what that meditation data was. Okay. This is not our data, somebody else collected that data, but it actually reveals something very interesting about gamma waves. So what is that? So what you are seeing here are four traces corresponding to data either recorded from control subjects or three different types of meditators. Okay, and I'm only showing you power here up to 30 hertz. I'm hiding the gamma frequency for now. You see that there is a bump at around 10 hertz or so. This is the alpha rhythm. So nice bump in the power spectral density. Some vipassana meditators have more alpha than others. What about the gamma? Actually it looks like this. Okay, so the control subjects have less gamma than meditators. All three types of meditators have more gamma. But notice that there is no bump in the power spectral density. So there's no gamma rhythm, so to speak. This is an elevated power over a fairly broad frequency range. So if you were to filter the signal in the gamma range, you will see more power in the gamma. But actually it's wrong to call it a rhythm, because it's just an elevated power over a broad frequency range. So this is what we'll call broadband gamma. And at this point you actually may feel that, hey, things are not right. Hey, everyone talks about gamma rhythms and gamma oscillations. So is there a case where you actually find gamma rhythms, a bump in the power spectral density in the gamma range? Gamma, by the way, is between 30 to 70 hertz. And turns out you can. There is a condition in which you will get nice bump in the power spectral density, but you have to work a little hard for it. You don't get it for free. What you have to do, it's actually very surprisingly simple. All you have to do is look at images like these. This is what is called a grating. This is a fixation dot, and if you look at this stimulus, your brain produces gamma waves. In the occipital area where the visual cortex is, you see that the signals start to oscillate. And this is a true rhythmic signal. So this is spontaneous brain signal. This is stimulus driven brain signal. This is data recorded from a monkey. And now in the power spectral density, you see a nice bump in the gamma range.
So independent of spatial frequency? Or
no, it depends on the spatial frequency, temporal frequency, orientation, size, all those things. And that's actually the key part of my talk, that you can control these gamma waves by controlling the stimulus that you are showing to these subjects. Okay, so I'm going to show you plots like these throughout the talk. You'll either see the power spectral density with a bump, and this other trace is before the stimulus was presented. I sometimes will show you a change in power spectrum, which means I'm just subtracting these two. The subtraction is done on log scale, so it's actually the change in power relative to the baseline period when there is no stimulus. This shows a bump in the power spectral density as well. I'm also going to show you time frequency plots. In this case, the stimulus was shown for 800 milliseconds. And notice that as soon as the stimulus comes on, the gamma rhythm is generated. It's a beautiful, bright, nice gamma rhythm. And once the stimulus is taken off, gamma rhythm goes away. Yes?
So what frequencies do you call gamma?
30 to 70 is the range. We'll see that you can actually make it vary, even go down to about 20 hertz or so. Okay, so there's that stimulus induced gamma. Now, as he was asking, does it depend on what kind of stimulus you show to the monkey, to a subject, monkey or human or cats for that matter? And the answer is yes. So you can control these gamma waves by showing different types of stimuli. In this case, if you show a small stimulus, you get a weak but fast gamma. If you make the stimulus larger, it's a slower but stronger gamma. So you have to think of gamma, the stimulus, as a hammer. Okay, so a little hammer produces a weak gamma, a big hammer, produces a strong gamma. Similarly, if you increase the contrast of the stimulus, gamma becomes faster. It's like having the same sized gamma, but hammer, but you're hitting it harder and harder. That's the equivalent of increasing the contrast, and gamma becomes faster and faster. Okay, so electrical engineers love this. This is, like you have inputs that you can manipulate, the signature, which is the output, which gets manipulated in very nice ways, and then you have got the transfer function, which is our brain, which we are trying to figure out. So recently we found many weird things about gamma. This is one of them. We were showing a monkey natural images and found that every time monkey looked at a tomato, we got massive gamma waves. These are even stronger than the achromatic gratings that I showed you earlier. Huge gamma waves. And then we asked whether, hey, it's the tomato or the color of the tomato. So we showed tomatoes in different colors. And look, only the reddish hues produce these gamma waves, other colors don't. And we still don't have a good idea why that is. In fact, you don't need a tomato at all. You can just show plain hues. So the screen full of just red or green or blue, and this is done in 3 monkeys here, all show very nice gamma only for certain colors, not for others. And we still don't know why that is the case. Okay, so that's all cool. Gamma depends on stimulus properties. So why should we care? Why is it useful to study these? Now, it turns out the gamma oscillations are thought to be associated, generated by an interplay of excitatory neurons. Here, these are the principal cells, and inhibitory neurons. In fact, the classical gamma that I talked about is thought to be associated by the interplay of excitatory neurons and one class of interneurons called the parvalbumin positive interneurons. Now there is a talk that another class of interneurons called somatostatin neurons produces a slower gamma, which is actually in the beta range. So inhibitory neurons actually are associated with a lot of different sort of mechanisms in the brain. In fact, if you have a mental disorder, often it's that the excitatory inhibitory balance is altered. So it's a nice readout. Gamma rhythms are a nice readout of the excitatory inhibitory interactions that are going on in the brain. And it's non-invasive, so it's in one way allowing us to study how excitatory inhibitory interactions take place in the brain. Okay, so that's why we think gamma oscillations are so interesting. Now, to take this a little bit more, think about it a little bit more abstract, in a more abstract way, we have here very famous David Marr's different levels of analysis, where you can think of, let's say here's a bird that's flying. So the flight is a computation that the bird performs, and there's an algorithm, in this case the flapping of the wings, and then an implementation, in this case how the feathers are implementing a mechanism that allows the bird to flap. So you can break down a particular computation in these different levels. In neuroscience, you can similarly think of behavior somewhere in the top, where I'm trying to figure out attention, decision making. I'll put mental disorders also here because it's a disorder in cognition, meditation, etc. and these levels. And then there are certain mechanisms in the brain that mediate these behaviors. So for example, attention you can think of as a gain control mechanism. Or in fact, in recent years, there's a particular mechanism called normalization, which I won't get into, which has been associated strongly with attention. And then there is a circuit level implementation of these mechanisms. So gamma oscillations have often been associated with actually a causal sort of, some people think that it's playing a causal role, some very important functional role. So it's with somewhere here mediating some mechanism in particular way. But we have a more modest view. We think that it's just a reflection, albeit a very sort of nice, interesting reflection. It's not playing a role per se, although we can't rule that out. But nonetheless, these oscillations actually nicely capture behavioral modulation as well as modulations, some circuit mechanisms that you can manipulate by manipulating the stimulus. And also there are lots of circuit level implementations of these mechanisms. So we can actually make associations between all these 3 levels with gamma oscillations, therefore allowing us to actually make a bridge between circuits, mechanisms and behavior. It's a complicated way of looking at things because we are all mapping it to something else here, but I'll give you lots of examples to see how we are trying to make this bridge. Yeah.
Is there any auditory stimulus which also?
Oh, we tried. Is Divya here? So there is one PhD student whose job was to find an equivalent auditory stimulus, and we didn't get it. So we tried a nice visual analog of the auditory stimulus, which is thought of like an analog of visual gratings. This is the stuff that Shihab uses a lot, ripples. They produce broadband gamma, not narrowband.
So there is no way where you can simultaneously have a high alpha and gamma?
High alpha and gamma, usually stimulus onset suppresses alpha and enhances gamma. So the type of manipulations that we do, which is only because of a stimulus, they go in opposite ways. One gets suppressed, the other one is enhanced.
You close your eyes and...
Yeah, we'll get to some of that when we talk about meditation data. Okay, so now let's move on. I'll just tell you, this is our sort of things that we do in the lab. We have got a monkey arm where we put, these are Utah arrays, but we also put other type of electrodes. These are ECOG arrays from AdTech, the company that, one of the prominent companies that puts these electrodes that are placed on the head of, on the brain of epileptic patients. So we have a setup where we simultaneously record spikes, local fields, ECOG and EEG from the monkey. So we can record from 4 scales in the monkey. And in humans, we record EEG. We also collaborate with neurosurgeons to record ECOG data here. So the goal is to actually make a bridge between the oscillations that you see in EEG down to responsive of individual neurons. Okay, so it's a cross-scale, cross-species setup, allowing us to actually understand in more detail, because we have access to neural data also, the relationship between oscillations recorded at the level of EEG and neuronal firing. And the type of questions that we ask span a lot of things, but neural oscillations, more particularly gamma oscillations, are always in the middle. In monkeys, we do a lot of studies where we show different types of stimuli and do a lot of coding, decodation and modeling and so on. In humans, we have a couple of studies where we have looked at the effect of aging, mental disorders, meditation and attention and so on. So this part is easier to explain. So when I have a half an hour talk, I usually just talk about the first half. When I have more time, which is the case today, I actually talk about, I'll talk about some of the monkey stuff as well, which I find very interesting, but it's harder to explain in some ways. Okay, so but we'll start with the easy stuff first. Well, it's not easy really, but it's easier to explain. So I'm going to talk about the effect of aging and mental disorders on gamma oscillations. This is all stuff done in humans. Okay, so why should we care about gamma oscillations and mental disorders? That's because, first of all, gamma oscillations are altered in a variety of mental disorders. Secondly, there's this new, well, it's not really new anymore, there's newish studies showing that if you can artificially induce this stimulus locked gamma, is different from the gamma that I was talking about. This is where a stimulus actually flickers at 40 hertz, like so, although it's not really at 40 in this case, producing what is called an SSVEP response. So it's a response only at 40 hertz. You show that to mice who have been injected with something that produces an Alzheimer's type phenotype. So their brain is full of amyloid beta plaques. Just viewing this light for a certain duration every day clears out this amyloid beta plaque. It's a Nature paper. Now there's a follow-up paper also in 24. Other papers that go against this, and so it's controversial in some ways. But nonetheless, this picked up, caused a lot of interest in the field, that hey, you can cure Alzheimer's disease just by showing flickering lights. And of course, you can buy that nowadays. So this is Li Hui Sai, who was the senior author in this. So you can buy this off Amazon for $25. So this light will flicker at 40 hertz. If you read the comments in that, in the review section, you will find, oh, people are loving it. They view it for an hour every day and say, oh, I'm very relaxed now and whatnot. Nonetheless, so we had a more modest way of looking at it. We thought that we will just show static visual stimuli, not the flickering ones, although we did try that as well, but I'm not going to talk about it. And we are going to simply see whether as you age, or if you have a mental disorder, whether your gamma oscillations are any different or not. So it's very simple sort of paradigm. Now to get started, we had to find a stimulus that produces very strong gamma in human EEG. Generally, all these studies are done in monkeys. You go invasive, you have a small stimulus that produces, that excites the neurons that you are recording from. But in this case, we wanted a good response at the level of EEG, because finally we were going to do that study in the human. So we first thought that we are going to try and find the best stimulus that we can get in monkeys, because the same monkey can be shown lots of different stimuli for days, and then we can pick out the best one that we will then use on elderly subjects. So that's what we set out. So this is our setup at Primate Research Lab. The monkey sits in front of a screen. We record using Utah arrays. This is from primary visual cortex. So my first PhD student, Vinay, and a project assistant at the time, Pujya, both of whom are now in Chicago, they were part of this project. And all the monkey had to do was look at the screen, look at this dot at the screen and not move her eyes while we show these large gratings. The only difference between our study and the previous ones is that we used full screen gratings, because we thought that if you have a large stimulus, then there will be more excitation and the EEG response will be stronger. That's the only reason we used that. And then Murthy, who was also a PhD student at the time, he would do the same to humans. They would put an EEG cap and look in front of a screen while we show full screen gratings, and that's it. Then they go home after a while and we get their EEG responses. So what we found is actually quite interesting here and very surprising to me. So here we are showing, in this case, a monkey, different stimuli at different orientations. And I should have mentioned this, I sort of forgot. You see this red circle here. This red circle is what is called the receptive field of the neurons that we are recording from. Just go back here. So the electrodes that we insert only care about a small part of the visual space. So they actually, the neurons don't care about what's here, 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 and so on. They only care about what's inside this circle. And as you record from other brain areas, these receptive fields become large. But in the primary visual cortex, the receptive field is very small. So that's why people in the past have only used small stimuli, because that's the one that the neurons care about. They don't care about what's elsewhere, so then why bother? But we nonetheless used full screen stimuli, and what we found is that you see this nice gamma rhythm, which is orientation tuned. We are not the first ones to find it. Many people have shown that gamma is orientation tuned. So in this monkey, horizontal gratings produce fast gamma, vertical gratings barely produce any gamma. But what we were surprised to see is that there is actually a second gamma. You see this? And it's tuned to a different orientation. So this is not some signal processing mishap where, just the harmonic of the rhythm is thought about as a different rhythm. It actually has a very different characteristic. It is strong when for a different orientation. It sort of builds up slowly over time, as opposed to fast gamma, which is set up within 100 milliseconds of stimulus onset. So we have two gamma waves, both in monkeys and also in humans. In human EEG, it's actually very hard to get gamma, but we got, after manipulating our stimuli properly, very robust gamma. It's a very nice looking, very reliable, strong signal. As soon as the stimulus comes on, you get this right spot, and it's localized to the back of your head. So this is what is called a topo plot. So looking at the head from the top, this is the nose, these are the ears, and this is the back of the head. So you can say gamma oscillations very strongly induced in humans while they are viewing the gratings. So we were wondering why others sort of missed it. Why not? Others did not report the slow and the fast gamma. And the reason is, when you show a small stimulus, you only get this fast gamma. This slow gamma, this other gamma that emerges, this is in two monkeys, you can see this emerging here, also in humans. This comes only when the stimulus is large. So a small stimulus produces fast gamma, large stimuli produce both gamma waves. And for reasons that I am not going to get into at all, we think that the fast gamma is produced by the parvalbumin positive network, and you can think of as a local processing gamma, while slow gamma is produced by another class of interneurons, which gives a more global modulatory signal. Again, why I am telling you this, I won't get into it. The proof is actually not as strong in primates anyway. It's mostly coming from rodent literature. Okay, so now let's look at the effect of aging in humans. What happens to gamma oscillations when people age? So this was actually a big study which went on for several years. Professor Ravindranath, who was the chair at the time, she was heading this project. Many faculty from Center for Neuroscience and other departments were also involved. So we recruited healthy subjects over 50 years of age from the community, and a bunch of people did a bunch of tests on them, including a lot of these clinical diagnostic tests. So this was carried out by Dr. Naren Rao. And then we, by we I mean Murthy and Kirtana and Santosh, he was a project assistant, he was a PhD student. They recorded EEG. And a very simple task, the subjects come, elderly people in this case, not young ones, they look at the screen, we show them greetings, and that's it, and they go home. We recorded from almost 280 subjects over a span of 3 years, 350 sessions. Later we found out that most of them were healthy, but some of them had preclinical Alzheimer's disease, mild cognitive impairment. At the time of the recording, we didn't know which one was which. Later we found that there are about 20 subjects with some sort of cognitive deficit. And about 50 subjects we recorded twice. Okay, so let's look at, first of all, just the healthy people, now separated into different age groups, and ask what happens to gamma as you age. So this is the first data set. The males here on top, females at the bottom. The color scheme is different. Here yellow means more gamma, this blue means alpha suppression. So females have more gamma and more alpha than males. It's actually significant result. This is actually a young cohort. This is not the cohort that I was talking about. The old cohort is between 50 to 90 years. So I'm separating them into two groups. This is the mid-aged and this is the elderly. And look at what happens as you age. So gamma becomes slower. Even if you are healthy, gamma oscillations become weaker and slower as you age. Okay? So, well, that's it. I mean, brain changes in certain ways. Maybe the inhibitory circuits that are generating this are no longer as robust. So, gamma will weaken, both slow and fast gamma, as you age. What about MCI, mental disorder? What happens there? So, as I said, there were about 20 subjects. For each of these subjects, we found the age and gender matched healthy controls. So we have lots of controls in this case, very few actual cases. So if you look at the gamma of the healthy controls of the MCI and AD group, that's good, strong gamma, both slow and fast gamma, very strong. What about the cases themselves? Very surprising. Their brains don't produce much gamma. Okay, it's a very strong reduction in the stimulus induced gamma. Alpha is about the same. These tick marks here as a function of frequency indicate the frequencies where there's a reduction, and you can see there's a reduction mainly in the gamma range. Yes.
In the previous case, when you had the healthy subject, you said the gamma was getting weaker in general, and now you say that the controls, which are healthy, they have strong gamma, but I suppose it is relative.
It's relative, yes, yes. So these controls come from different age groups. So within the controls also, as they age, you will see a reduction, but it's stronger compared to the age and gender matched cases here. Okay, so it's a strong reduction. So mental disorders like Alzheimer's disease reduce gamma oscillations. Now you might ask, how reliable is gamma? Okay, so if I show you gamma the gratings, and then you have a particular gamma signature, and if you come back later and I do it again, will it be reliable or will it be very different? So turns out across individuals there's a lot of variation. These are 20 individuals, they are all healthy as per the this clinical tests. They all have different gamma patterns, so to speak. Somebody has one prominent wave but not a strong not a strong fast gamma. Some people have this one wave, another one that comes like this. One subject has very fuzzy looking gamma. So there's a lot of variation across subjects. But we recorded from about 50 subjects twice, separated by 1 year or more. So we have done hundreds of recordings between these two epochs. They come back a year later, we do this again, and we find that it's actually very similar. See, if you look at this pattern and that pattern, this pattern and this pattern, that pattern and that pattern, people who don't have gamma, there are some people who don't show much gamma, continue to not show gamma. You can actually identify a person. These are all male subjects, these are all female subjects. So you can see that this signature is very reliable for a given subject. Okay, it varies across subjects a lot. It's like a fingerprint of your brain. Everybody has a different gamma signature, but it remains consistent for that person. You can actually do a sort of classification analysis and get ROCs above 0.9. So you can identify a person by their gamma signature. So that's good. It's a strong, robust, reliable signal which varies from person to person, but it's very robust for a given person. Okay. So
from which electrodes is
these are all occipital electrodes. So gamma is very strongly concentrated in the occipital electrode, in the visual cortex.
And is it very sensitive to the placement of the electrodes, or not so much? This would suggest not so much.
Well, so these are all recorded.
Exactly the same stuff.
Yeah, but so we have a cap, it's a brain products cap. So once you put it, it's more or less in the same location, but it's not that sensitive. So you'd see good gamma in about 8 to 10 electrodes in the visual, in the back of your head. We'll look at the average of those. And sometimes one electrode doesn't work, and it really doesn't matter that much. You get a robust signal if you take an average. Okay, one more thing, and then I'll move to meditation, and then I move to the monkey stuff. So this one last thing is actually related to, not to gamma oscillations, but actually to another feature, which is the slope of the power spectral density. So this is the power spectral density. This is not gamma, this is line noise at 50 Hertz. So you will find these pointy things, they are boring and uninteresting, pay no attention to those. The slope of this power spectral density for mid-old versus old, you see that it becomes a little bit shallow. We are not the first ones again to report this. Way to think about it, we actually have a more detailed story here. I won't get into that. We looked at the slope of the power spectral density up to 1000 Hertz and found some interesting differences there, but I won't get into that. The way to remember is, why this is happening is, in general, this is something that we'll keep coming back to, that excitation is fast and inhibition is slow. So if there is more inhibition in your system, then it's going to wipe out the high frequency content, because inhibition is slow, it's like a low pass thing. So it will keep the power spectral density steep, and as your inhibition wanes away, higher frequency components will increase, so it will make the spectral density more shallow. Okay, and in the meditation data this will become important, which is why I'm showing it here. As a person ages, not only does the stimulus induced gamma weaken, but also the power spectral density slope becomes more and more shallow. Okay, so we are actually very big on making this sort of data publicly available and useful. So all the data, raw data, as well as all the codes for all these projects is actually publicly available. So you can go to GitHub and actually regenerate any of the figures from any of these papers, starting from the raw data. So we think that this sort of sharing will allow us to make our results better, and others can look at other features that we haven't looked at. So all of this is freely available. So if you like to study gamma oscillations in EEG, you actually have the data freely available. Okay, now I'm going to switch gears and talk about meditation. Figured it's fun to do meditation studies. In India, you have a rich tradition of meditation. You have many different types of meditators. So we should do a meditation study. And we actually have a name for that project. It's called Project Dhyan. Dhyan means meditation in Hindi and Sanskrit. So Ankon, who's sitting here, and Srishti, who's also a PhD student, not sure if she's here, and Dr. Kanishk, who was a postdoc and now is an assistant professor, these were part of this study. We were also helped by Dr. A.G. Ramakrishnan and his students, Pradeep and Akash, earlier part of this study. So what did we do? Meditation has been studied for a long time, as you all know. People usually get meditators or their age and gender match controls and ask them to meditate. And before they meditate, you have some control conditions where they just keep their eyes open or closed. And this is what people have done for like 50 years or so. So nothing new in this. This is a typical meditation study. What we did is that we added these two conditions. This is what we call G1 and G2. A gamma protocol before they meditate and a gamma protocol after they meditate. In these protocols, they just stare at the screen while we present the gratings that induce gamma waves in their brains. So the question is, do meditators have stronger gamma than their age and gender matched healthy controls? Now obviously before meditation you can study gamma, after meditation you can study gamma. During meditation can you study gamma? Typically not, right? Because their eyes are closed. How will you induce the gamma waves?
How did you show the gratings while eyes were closed?
That's what. So in G1 and G2 the eyes were open. So you can show this, but during meditation you typically can't. But again, this is where, as I said, we have such a rich tradition of meditation type, there's so many forms of meditation. So we found one form of meditators called Brahma Kumaris who meditate with open eyes. So their eyes are open and they are meditating. It's awesome. So because of that, we are able to actually add another protocol here. While they are meditating, we call this the M2 protocol, with open eyes while we are showing the gratings. So now you can study gamma before meditation, gamma after meditation, but also gamma during meditation. So, okay. Now, other than just looking at EEG data, we also wanted to look at the physiological parameters. We have actually not analyzed any of this at all, but we have recorded heart rate and respiration rate and eye movements. So we have a very fancy eye tracker with a sticker on top, so that even when you don't you move a little, you can track your eyes. And we going forward, we want to actually study how these different physiological parameters associate with the brain waves. For now, we haven't done any of that. So I am going to tell you 5 interesting stories in, let's say, 5 minutes. Quickly we will go through this because I want to spend 15 minutes at least on the monkey stuff. So let me just very briefly tell you the first set of findings that we have from this study. So let's look at first condition when their eyes are open but they are not meditating. Okay, this is just open eyes, and simply ask, because of this long meditative practice which went on for, like these are all very expert meditators, is their brain somewhere different, so that even when they are not meditating, is there a difference in the brain signals? So turns out there is. So these are occipital electrodes, back of your head, but I'm also going to show you the frontotemporal electrodes. And for these sets of electrodes, what we are doing here is showing the power spectral density of the controls and meditators. Remember, there is no stimulus here. They are just keeping their eyes open. By the way, we track their eyes, so if they break fixation and those things we take care of. Okay, so if they blink or break fixation, then the trial is aborted. We have control for eye movements very well. So generally, meditators have more power at higher frequencies. This is the broadband gamma. You see that there's an elevated power in meditators versus controls, even when they are not meditating. Okay, so they generally have more broadband gamma, which is more prominent in the frontal part. Interestingly, if you now look at the slope of the power spectral density at higher frequencies, this is the slope. So this dotted line is the slope for controls. This is for meditators. You see that the slope is actually slightly steeper for meditators than controls. So remember what I told you, as you grow old, spectral density flattens. Now here meditators and controls have the same age, but the meditators have steeper spectral density. So in some ways it's going against the effect of aging. It's like reversing that because they have a steeper power spectral density, also suggesting that they have a more robust inhibitory network. Because as I told you, inhibition is slow. When it's active, it actually diminishes high frequency components and steepens the power spectral density. Even when their eyes are closed, we see the same result. So it's not some eye movement artifact and things like that. What happens when they meditate? Remember, again, now I'm going to show you meditation data when there is nothing on the screen. So again, this is just to reconfirm what others have done. So far there is no grating in the picture. So they are meditating, and I'm going to compare the data while they are meditating with open eyes versus when they just have open eyes but they are not meditating. So this is what you find. Even both in occipital and temporofrontal electrodes, the dotted lines show the power spectral density while their eyes are open but they are not meditating. And in color, solid lines show when they are meditating. And you see there's an increase in the power. This power actually goes up. It's more in the frontotemporal electrodes. So the whole power spectral density shifts up. This is the original paper that I had showed you, that when people meditate, the power spectral density increases. It's more prominent in the frontotemporal electrodes than occipital. It also occurs for controls, but only in the frontotemporal area for controls, not the occipital ones. In meditators, it's occurring in both. Okay, we'll see a more direct comparison when I get to M2. So there is broadband stimulus free gamma, which increases in both meditators and controls, but in meditators it increases everywhere. In controls it's more localized to frontotemporal regions, and it's a broadband gamma. There's no stimulus in picture. Now comes the killer question. This is the one that we were after. So what happens to stimulus induced gamma waves? Okay, when they are looking at a grating before they meditate and after they meditate, what happens when to the stimulus induced gamma? And this is where we actually are very happy to see that while the alpha is unchanged, so this is change in power, so you can see that there's these tick marks mean a significant difference. So alpha is unchanged and fast gamma is unchanged, but look at this slow gamma. It is much stronger in meditators than controls. Again, if you look at the topo plots, it's a much stronger gamma in meditators than controls. Also in the time frequency plot, much stronger slow gamma in meditators than controls before they meditate as well as after they meditate. So their brains have somehow changed over a time, this long duration of meditation that they've done for years, which somehow leads to a much stronger slow gamma rhythm. Okay? Again indicating that they may have a much stronger inhibitory network which is producing these gamma oscillations. Finally, because we have this condition where they have their eyes open while we are showing the gratings. Remember, the gratings are shown for 1.25 seconds and then there is a blank period. It keeps alternating. So you can study the stimulus induced gamma during the stimulus period, also the stimulus free gamma during the spontaneous period. You can measure both. And what we find, that the stimulus free gamma, the spontaneous gamma, this broadband increase that I was talking about, is stronger in meditators than controls, and it's in the frontal part. How do you remember that? You remember as if there's a third eye here. The meditators think of, actually these meditators are focusing on their forehead. So as if the third eye is sort of causing this elevation in the frontal region, which is stronger in meditators than controls, while the stimulus, when it comes on, produces a stimulus induced gamma, which is also stronger in meditators than controls, but it's in the occipital area. So you have both stimulus free broadband gamma and stimulus induced narrowband gamma, which is stronger in meditators than controls. Okay, because you have both, we tested if there is a correlation between the two and we didn't find one, so they seem to coexist but are independent.
Is the alpha suppression same for both?
Alpha is not different. So alpha is unchanged. You don't see a peak here. It's only very selective to slow gamma. It's actually nice because some people say, hey, maybe there's a difference in here and, other things, lifestyle. So again, if those are there, then you would see a difference over a large frequency range. Here it's a very limited frequency range where you see the difference. Okay, so I'm going to summarize that gamma oscillations are cool. And meditation could be boosting the long range inhibitory network, the somatostatin network, and potentially provide protection against aging and neurodegeneration. But we have to do a lot of work before we can establish this. Okay, I'm going to now move on to a different question. Okay, any questions on this? Or maybe we can do this after 15 minutes so that, or what shall we do?
It's up to you.
Okay, let me just finish this because I don't want to miss out on this. This is stuff that's very close to my heart. So I'm going to talk about this quickly and then we'll take questions. Okay? Alright, so neural oscillations, gamma oscillations and binding problem. This is the thing that in some ways started it all when it came to gamma oscillations, because a guy called Wolfsinger found a very interesting had a very interesting finding which was associated to this question of binding. So what is binding? Well, so the binding problem is as follows. Here's this is cricket here, batsman hitting a shot. So we are looking at the ball as it flies away through our visual space. Imagine you sitting there in a stadium looking at this. The fact that, so suppose you're looking at the ball, so the fact that it's round in shape, the fact that it's white in color, the fact that it's moving in a certain direction, all these attributes are actually processed in different brain areas in the visual cortex. There's one area that processes color, another area that processes speed, But you never have this feeling that, oh, there's a lot of stuff going on. You always have this unified feeling of a ball flying through the visual space. So somehow all these features that these neurons are encoding, they need to be bound so that you have this unified feel of a ball flying through the visual space. So who's doing the binding? That's the binding problem. It's a deep philosophical question. So Wolfsinger, it's a very seminal, very classic paper, showed something that may shed light to this. So what he did was that he put two electrodes, this is a cat here, which have these two receptive fields. The receptive fields are here and here. So the neuron that I'm recording from this area only cares about what's in this rectangle. The neuron in this area from this electrode only cares about what's in that rectangle. And he showed a long bar and moved it back and forth. Okay? And when he did that, if you look at the cross correlation between these two neurons, you see a nice oscillatory pattern at gamma frequency. So you see that the cross correlation. So the neurons are in this case firing in synchrony at 40 hertz, at around 40 hertz. Now we introduced a little bit of a cut here. Now the cut is outside the receptive field. So as far as these neurons are concerned, they shouldn't care about this cut at all, because they were seeing a bar before, they are seeing the same bar in their receptive field now. And the firing rates actually don't change, but what changes is this cross correlation function. This synchrony between the two neurons reduces. And somehow when there's a long bar, so you need to bind the features of these two bars, segments, so that you know that it's one unified bar. The fact that gamma synchrony has reduced made them sort of hypothesize that binding is achieved through synchrony. This is the binding by synchrony hypothesis. Okay, so gamma oscillations are actually the ones that are synchronizing the population and causing the features to bind. So by the way, it's a very controversial hypothesis, and as we will see that we may have simpler answers that may not have to go about binding. But let me go back now and tell you about what we did here. So see, these are the two ends that Wolfsinger studied. A continuous stimulus produces strong gamma. It was a bar before, now it's a grating, but the idea remains the same. And a discontinuous stimulus produces weak gamma, right? Now nobody has actually manipulated the level of discontinuity. What happens where it's only a little bit discontinuous, that's like a small gap here versus a condition like this? What do you expect? Well, one possibility is that as the discontinuity increases, gamma oscillations progressively become weak. That's one possibility. Another possibility is that gamma acts like a sort of like a resonance model. Even a small discontinuity is enough to break gamma rhythm. Okay? Nobody had done it, so we thought, hey, let's do this. Seems like a fun study to do. Any sort of show of hands, who thinks it should go like this? No one, eh? How about, if nobody raises hand, that means everybody is sleeping. Who is for the population model, where there's a graded decrease? Okay, who's for the resonance model? All right, and who's sleeping? All right, so this was actually the first study that we did in the lab, one of the first studies. This was Vinay's PhD thesis. So monkey comes and sits in front of a screen. We measure it, find out where the receptive field is. We either show a big stimulus or a stimulus with a discontinuity. And we actually had many different types of discontinuities. Discontinuity in space, in orientation, in phase, in contrast, and so on. And what do we find? Big continuous stimulus, lots of gamma. Discontinuity, very little gamma. As I reduce the discontinuity, will gamma increase progressively? No. Even a small discontinuity, it's like a hairline fracture in the stimulus. Can you guys even at the back even see that there is a discontinuity here? It's a very small discontinuity, it completely wipes out gamma oscillations. So gamma is a very fragile signal. Your stimulus has this and that, you won't see it. You have to really work hard to get this strong oscillatory signal. Small discontinuity is enough to kill gamma. Here again, there's only a 10% orientation difference between the center and what is called the surround, and it almost completely wipes out gamma oscillations. Very surprising. I was putting all my money on the progressive thing, but this is what it is. And you can say, hey, there was some change, so can I compare with, let's say, the firing rate? So this is the firing rate data. I'll just show you this, that the change in gamma power when you have a discontinuity is a lot more than change in firing rate. So there's some increase in the firing rate, but the reduction in gamma is a lot more than the increase in firing rate. So here we have a system where the firing rates have barely changed with the introduction of a discontinuity, but gamma is wiped out, almost wiped out. Yeah.
Were the neurons recorded in the periphery or close to the fovea?
So the neurons were about 3 degrees from the fovea. The foveal receptive fields are so small that you can't, little bit of eye movements just wipes it out. So we tried to keep it between 3 to 4 degrees, which is what was the case in this case. Okay, so now that's cool. We have a interesting sort of observation. How do we make sense of this? And this is now getting from mechanisms to circuits. Can I build a circuit that shows this? Okay, and we'll see that a very simple circuit can actually explain this. So what is that circuit? Which is what is called a Wilson-Cowan model. It's a very famous model. Almost 50 years ago, Wilson and Cowan built this. There's one excitatory population and one inhibitory population. And the excitatory guy excites itself and the inhibitory guy inhibits itself and the excitatory guy, and they both get these inputs. And you can write down the firing rate of the excitatory population Re and that of the inhibitory population Ri using these two differential equations. Okay. Now here GE, this whole thing within the bracket is the total input that the excitatory population is getting. It's WEE multiplied by E, Re minus WEI multiplied by Ri plus the external input. So this is the total input. This function, which is called the activation function, converts the input to firing rates, and then this model essentially tells you how the firing rate should change. So these are differential equations. How do we make sense of this? One easy way is to just ask, when is this derivative equal to 0? When is dRe dt equal to 0? So that means Re is going to be equal to this. So you can simply find out the values of Re and Ri for which this derivative is 0. So those values constitute what is called a null cline. So the excitatory null cline would look like this in a space where the excitatory firing rate is plotted as a function of inhibitory firing rate. So if you are anywhere on this line, the excitatory population is very happy because its time derivative is 0. It can sit there forever. Similarly, you have the inhibitory null cline, which is all the conditions for which this derivative is 0. And whenever these two null clines meet, you have a point where both the derivatives are 0. So if you keep the system at that point, it will stay there forever because both the time derivatives are 0. These are called fixed points of this dynamical system. Now the question really is, what is happening if you keep the system close to that fixed point? And there are various possibilities. One is that the fixed point is stable. So if you are near it, you are actually going to spiral into it. And as a function of time, it's going to be a damped oscillator. There could be another place where the excitatory, the fixed point is not stable, but as the system goes near it, it is repelled by the fixed point but starts oscillating near it, generating what is called a limit cycle. So limit cycle in the time domain is an oscillation. Okay. So you can translate the null clines by changing the inputs. If you change IE or II, it's going to translate the null clines, and you can in some conditions go from a fixed point to a limit cycle. And this is equivalent of, so in this case you increase the inhibitory input by making the stimulus large. There is now more inhibition coming in, and the system will translate and generate stronger gamma oscillation. This is the increase in size increasing gamma wave. So you have to fiddle around with the weights and the activation function, but you can put it in the right spot so that changes in contrast, changes in size, etc. can be explained by this model. Which by the way, Jadi and Sanowski, Terry Sanowski and Monica Jadi did in 2014. So we have been following up on their work. But we wanted to do something else. We wanted to explain the discontinuity effect, right? So now how do we do it? Well, what we figured, so this is really a very sort of hack almost, a very simple trick. So what we said is that, look, in the brain you have the neuron, and it gets inhibition from many different circuits. So essentially you can think of these models within each cortical column, and they also interact with each other. So you have one Wilson-Cohen model, which is explained by this formula here, and you have another guy which is also explained by this formula here, but they interact. So there is lateral excitation and lateral inhibition from nearby guys, which correspond to these inputs. Okay. Now this is a sleight of hand, which you can see here. If you actually change the signs here, you essentially now have the total weight being parceled into two parts, which is, one is its own weight and the second one is the recurrent weight. Okay. And what we said, that if there is a discontinuity, so it wipes out one of these models, so that part is not getting excited anymore. So essentially it will cause a reduction in the weight of the Wilson-Cohen model. So discontinuity means a reduction in the weight in the Wilson-Cohen model. That's it. And surprisingly, this simple thing works. This simple thing works because we set up the model in a way that it sets up in an oscillatory regime in the beginning. So these are the weights. This dark brown is in the oscillatory range because it's a limit cycle. In fact, you can work out the math. This is what is called a supercritical Hopf bifurcation, and it is an unstable point here generating a limit cycle, so you get strong gamma oscillations. If you change the weights even by 10%, keeping all the other weights the same, it causes a reduction in gamma waves, and if you change it by 20%, it just wipes it out. What is happening is you are sitting on one side of the bifurcation point where it's a limit cycle, and this little bit of a change in the weight, which changes the slope of this null line, moves it from an unstable point to a stable point, and the gamma oscillation goes away. Okay, it's like steam at 100.5, you cool it a little bit, it becomes water. So gamma oscillations are a resonant phenomenon. Okay, they are very sort of, you can set it up, but they are easy to break, and a little bit of a change in the system can actually move it from an oscillatory regime to a stable regime, and you lose the gamma rhythm. Okay, now I have finished my time. There was something I wanted to show, but I guess I'll just stop here. It's been 1 hour and 2 minutes by my watch. Just to mention that actually even with a simple model like this, a Wilson-Cohen model, you can study a lot of properties of the system and actually find out very interesting things, like how the excitation and inhibition is changing. In fact, Krishna Kumaran, my PhD student, who's actually going to defend only on Friday, this Friday, he actually manipulated this model in interesting ways and showed some very interesting properties that explains its burst duration and the way the gamma rhythm changes over time and things like that. But in the interest of time, and because I see many of you get really tired now, I'll stop here. If you are interested, we can talk about this one-on-one later. Okay? So that's it, and these are the things, but I'll just leave you with this, that in our case, gamma is a readout. We build models to explain certain mechanisms like adaptation and discontinuities and contrast and size, hoping that one day the way gamma changes with high level behavior, even let's say meditation and so on, will give us clues about the underlying mechanisms that mediate high level cognition. Thank you very much.